Dear friends, this video is about quality aspects, their breeding prospects in different crops. Quality refers to suitability or fitness of an economic plant product in relation to its end use. Definition of quality varies according to our needs from the viewpoint of seeds, crop growth, crop product, post-harvest technology, consumer preferences, cooking quality, keeping quality, transportability, etc. Quality traits means a trait that defines some aspects of produce quality. Each crop has a specific and often somewhat to completely different set of quality traits. Classification of quality traits: morphological, organoleptic, nutritional, biological, and other quality traits. These traits are with respect to plants or field crops. Coming to the morphological traits, this is related to produce appearance, concerned with size and color of the produce, example, grain or fruit size, grain or fruit color, etc. Easily observable, usually play main role in determining consumer acceptance of the produce. Organoleptic traits. This is concerned with palatability of the produce, example, taste, aroma, smell, juiciness, softness, etc. This trait is easily detected, very important in influencing consumer preferences. Nutritional quality. This determines the value of the produce in human or animal nutrition. It includes protein content and quality, oil content and quality, vitamin content, mineral content, etc. Also the presence of anti-nutritional factors. This trait is not easily appreciated by consumers and farmers, but they are of paramount value in determining human and animal health. Coming to the biological quality traits. The traits included in this group defines the actual usefulness of the produce when consumed by experimental animals. This usefulness to human is usually predicted on this basis, example, protein efficiency ratio, biological value, body weight, gain, etc. These traits are not obvious to consumers and growers, but are extremely valuable in determining the utility of produce for human or animal consumption. The other quality traits include which are not included in the categories which are discussed previously. Example, cooking quality of different crops, keeping quality of fruits, vegetables, fiber strength of cotton, etc. Many of the traits in this group are of prime importance in determining the usefulness of concerned produce. Coming to the quality traits of selected crops. White or amber color grain, medium to bold size, hard vitreous texture and lustrous appearance are important features of good market quality. High lysine content and good baking quality are essential for use in biscuit and bread manufacturing. These two points are related to wheat quality aspects. Coming to rice, white colored fine and long slender grains, taste and fragrance, less breaking in milling, more hulling recovery, better cooking quality, high protein and lysine content. In maize, bold flint grains with attractive color, high lysine and oil and sugar content. The seed color should be yellow or white. In sorghum, bold, thin pericarp, white grains of attractive shape and size with high protein and lysine content are preferable quality characters. In pearl millet, bold, lustrous and pearly amber color grains with high iron content. In barley, malting quality, low protein content and high extract of soluble oligosaccharides after malting are desirable characters. Low protein produces less haze in beer and high oligosaccharides are suitable for fermentation. In pulses, the quality aspects are attractive shape, size, and color of grains with high protein content, high methionine and tryptophan with less flatulence. In oil seeds, the attractive shape, size, and color of seeds, high oil content, free from anti-nutritional factors, and more proportion of unsaturated fatty acids is important. The moderate hardiness, long intermediate optimum fiber for milling, sucrose ratio, high sucrose content, and good quality of juice are characters of sugarcane. In cotton, fiber length, strength, fineness, maturity, uniformity, and different color of the cotton for color cotton production are the quality traits. In tobacco, short and thin leaves with less branched veins are preferred for cigar. Thin leaves are also preferred for pipe smoking. Thick leaves are suitable for cigarettes. High nicotine content for BD, hookah, and chewing and low for cigarette are preferred. High sugar content is also preferred in tobacco. In sun hemp and mesta, length, strength, fineness, color, luster, freedom from knots and specks is important. In potato, attractive shape and size, color of tubers, taste, cooking quality, thin skin, keeping quality, high starch, and less greening. In vegetables, high vitamin, mineral content, good taste, keeping quality, and cooking quality. In forage grasses, greater nutrition value, more palatability, and freedom from toxic substances. In medicinal plants, high content of active substances. There are four major goals for breeding for improved nutritional quality. The first one is high content and quality of protein, high content and quality of oil, about vitamins, and low toxic substances which are harmful to human health. Coming to the nutritional aspects, the scientific study of food in relation to health is referred as nutrition. Various chemical components of food which provide nourishment to the body are called nutrients. These are carbohydrates, fats, protein, vitamins, minerals, and water. Good nutrition refers to adequate intake of well-balanced diet which supplies all essential nutrients required by the body. Malnutrition may result from deficiency, excess, or imbalance of nutrients. Hence, all the nutrients should be taken in adequate quantities. Protein content and quality. Proteins are an essential component of diet. These are organic macromolecules consisting of a long chain of amino acids linked with other peptide bonds formed by carboxyl group of one amino acid with amino group of other amino acid. The nutritional properties of proteins are determined by their amino acid composition. There are 21 amino acids which are important in human nutrition. These are classified into essential amino acids and non-essential amino acids. The chemicals synthesized in body and the requirement has to be met through dietary intake. There are methionine, isoleucine, leucine, lysine, threonine, tryptophan, valine, phenylalanine, histidine, and arginine, which are called as essential amino acids. Out of these, arginine and histidine are considered non-essential for the adult. The non-essential amino acids can be synthesized in human body and they need not be supplied through diet, like cysteine, cysteine, proline, glycine, serine, alanine, aspartic acid, hydroxyproline, glutamic acid, neuroleucine, and tyrosine. The quality of protein is determined by content of essential amino acids. The sulfur containing amino acids are tryptophan, threonine, Isoleucine, lysine, valine, and methionine are referred as limiting amino acids, mainly in case of pulses. Coming to some limiting amino acids in some food crops, cereals are deficient in lysine, threonine, sometimes tryptophan. Pulses are deficient in methionine and tryptophan. Nuts and oilseeds in lysine. Green leafy vegetables and grasses are deficient in methionine. Coming to vitamins, a vital substance which is required in very small or minute quantity and is essential for proper growth and good health is called vitamin. Vitamins are essential for body growth, maintenance and reproduction. Based on their solubility, vitamins are of two types. One is fat soluble and the other is water soluble. Fat soluble vitamins are A, D, E and K, whereas water soluble are B and C group vitamins. Vitamin D and folic acid can be partly synthesized in our body. Rest of the vitamins has to be supplied through diet. Vitamin D is found in fats. Other vitamins are found in fleshy fruits and vegetable crops. Coming to the nutritional quality of cereals and pulses. Cereals are important source of carbohydrates. The contribution of cereal is 70% to the total calories in human diet. They contribute 50% to the global protein requirement. While grain legume contributes only 20%, remaining 30% come from animal products. The average protein content in the cereal ranged from 10 to 13%. However, cereal protein is deficient in lysine, tryptophan and threonine. Coming to the cereal protein quality. Cereal proteins are classified as albumin, globulin, prolamines and glutamine. Albumins are soluble in water. These are balanced amino acids. Globulins are also balanced amino acids and these are soluble in saline. Prolamins are soluble in strong alcohol and these are not balanced because deficient in lysine, tryptophan, rich in proline and gliadin. These are the major fractions in wheat, barley, maize and sorghum. The fourth one is glutanins. These are diluting in alkali or they dilute in alkali and balanced amino acids. So by this table we can conclude that among the four cereal proteins, prolamine is imbalanced and this can be found in some of the cereals. In all cereals, prolamins are relatively rich in proline and glutamine but low in basic amino acids including lysine. Therefore, they have poor nutritional value. Generally, prolamine content in cereals is negatively associated with total protein content. Cereals contain about 70% prolamine and glutenin and 30% albumin and globulin. Several mutants with improved protein quality have been identified in cereals like maize, jawar and barley. Coming to the mutants affecting protein quality in cereals. In maize, OPAC2, OPAC7, Flori2, Flori7, Brittle2. These are spontaneous mutant. The features are they are with high lysine, tryptophan and sulfur containing amino acids. Flori2 is only mutant which has high lysine and sulfur containing amino acid, whereas rest of the mutants of maize are rich in lysine and tryptophan. The remarks about these mutants, these have reduced prolamine and modified starch reduced prolamine. In barley, the mutants are hiplori, that is spontaneous with high protein and lysine. This has also several minor genes. Notch 1 is and Notch 2 are induced by ethyl methane sulfonate, that is EMS, a mutant, potential mutant that made high protein and lysine content with reduced prolamins. RISO 1508, that was induced by EI for high lysine for reduced prolamin quality change in the proteins. In sorghum, IS 11167, that is a spontaneous mutant with high lysine but caused shriveled endosperm and reduced prolamin. P721 is an opaque which is induced by DES. Feature is high lysine and reduced prolamin. Monogenic modifying genes make endosperm vitreous. P271 is vitreous and that has reduced prolamin. Coming to the second class, that is legume protein improvement. Legumes contain protein from 18 to 28%, which is almost double of cereals, except soybean, which has 43% protein. In pulses, two types of proteins, albumins and globulins, are found. The major storage protein in grain legumes is globulin, which constitutes about 80% of the total seed protein. In chickpea, globulin is of three types. One is alpha, then beta and gamma. Alpha globulin accounts for more than 80% of the total protein. In faba bean, pea, globulin is of two types, that is legumin and vicillin. Legume is less soluble in salt solution than vicillin. Moreover, legumin does not coagulate at high temperature while vicillin coagulates. Protein legumes are deficient in methionine and tryptophan. Moreover, they contain several toxic substances like protease inhibitors, hemagglutinin, lethyrogens, glucosides, goitrogens, cyanogens, metal binding factors, and antivitamin factors. However, many of these toxic compounds are destroyed during cooking. Some of these toxins have to be eliminated through breeding. The mixture of cereals and pulses is 3:1 ratio, provides nutritionally balanced diet. The protein fractions present in legumes. First one is albumin, which is water soluble, balanced, and minor fraction is 20% or less. Globulins are soluble in saline and deficient in sulfur containing amino acids, but predominant fraction is 80%. In chickpea, the protein fraction is somewhat different. Here, alpha globulin and beta and gamma globulins are found. The balance is these are deficient in sulfur containing amino acids, whereas beta and gamma globulins are balanced. The alpha globulins are 80% of total globulins, and beta and gamma, which are balanced, are relatively with minor fractions. The next is pea, faba, and French bean. In legumin, less salt soluble, non-coagulant. These are about balanced, more predominant than in vicillin in faba bean. Vicillin is more salt soluble, coagulates at 95 degrees Celsius, which is deficient in sulfur containing amino acids, and almost the only globulin in French bean. Genetics of nutritional trait. The quality traits are governed by oligo, poly, and maternal effect. Coming to the oligogenic inheritance. Inheritance governed by one or few major genes. Each gene has large and easily detectable effects on the expression of nutritional quality character. The differences between characters of high and low value is clear cut. In sorghum, high lysine content is controlled by a single gene with incomplete dominance. In barley, high lysine content is governed by one major gene plus several minor genes. In safflower, fatty acid composition is governed by one major gene with three minor alleles. In tomato, high beta carotene content is conditioned by two major genes plus modifiers. Polygenic inheritance. This is governed by several genes each with small additive effect. In such inheritance, the variation for a character is continuous from one extreme to another. Classification of plants into clear-cut classes is not possible. The protein content in cereals, pulses, and oilseed content in oilseed crops are governed by polygenes. Even the disease, I can say in broad terms, the biotic and abiotic, many of the factors are also governed by the polygenes. Characters which are governed by polygenes are sensitive to environmental changes and generally have low heritability. In carrots, high carotenoid content exhibits complex inheritance pattern. Coming to the third one, maternal effect. This is important or present in some quality traits. Usually such traits are concerned with grain characteristics, example seed size, protein content, etc. Example reported for protein content in chickpea, fatty acid composition in maize, soybean, rapeseed is influenced by polygenes or genotype of maternal parent. Maternal effects have same effect on genetic advance under selection as other environmental factors, like they confuse correspondence between genotype and phenotype and thereby reduce the progress under selection. Therefore, selection schemes must have make allowance for maternal effect and if and where present. The genetics of nutritional quality characters of some crops. In sorghum, quality character is high lysine, that the inheritance is controlled by single partially dominant gene. In barley, high lysine is governed by one major gene and several minor genes. In oats, protein content is governed by complex low content is dominant over high content. Maize, sunflower and safflower, seed oil content is additive gene controlled. In sesamum, seed oil content is additive genes with partial dominance for low oil content. In rapeseed, erucic acid and eicosanoic acid controlled by two genes with multiple alleles. Turnip rape, quality characters are erucic acid and eicosanoic acid. These are anti-nutritional factor and these should be removed or reduced in rapeseed, turnip and other brassica groups. Here, single gene with multiple alleles are governed. In safflower, fatty acid composition, three major alleles at one locus. In tomato, high beta-carotene content controlled by two major genes plus modifiers. In carrot, carotenoid content, the inheritance is complex in carrot. Coming to sources of quality traits. The sources are cultivated variety, germplasm line, spontaneous or induced mutant, somaclonal variant, wild relative and a transgene. Coming to a cultivated variety. This is a most preferred source. Example, Atlas 66 and Nuffle have been used as source of high protein content in USA. A germplasm line, example high lysine, more 2-3% of total protein lines of sorghum, that is IS 11167 and IS 11758 were identified from Ethiopian collections. These lines also have 15% protein, but their seeds are shriveled and red in color. They have been extensively used in breeding programs. Third one is a mutant. Many quality traits have been contributed by spontaneous or induced mutants. There are also examples of isolation of desirable mutants from mutant lines for quality traits. Example, P721 opaque mutant of sorghum has opaque endosperm which is not liked by consumers because of its shriveled nature and the bad color. A witch's endosperm DES induced mutant was isolated from P271 opaque line. This mutant has high lysine content and accepted one. The fourth one is a somaclonal variant. This may sometimes show an improvement in quality trait. Example, some clonal variant of sweet potato had a deeper and more stable root color, which is preferred by consumers. This variant was released as a new variety called Scarlet. A wild relative. There are several instances where genes for improved quality were contributed by a wild relative. In many cases, the quality trait is not expressed as such in the wild species, but it is detected only in the segregants recovered from its cross with a cultivated relative. Such traits are called latent traits. Example, Letha hirsutum, a wild relative of tomato, that is Lycopersicum hirsutum, produces small green fruits. Yet some of the lines extracted from a cross between cultivated tomato, that is Lycopersicum esculentum, into Lycopersicum hirsutum showed enhanced red color, while some others showed considerably higher carotene content. Here is a list of quality characters transferred from wild relatives. In cotton, Gossypium thurberi, the quality trait is fiber strength. Gossypium armorianum for fiber strength. Gossypium enamelum for fiber fineness, strength and maturity. Gossypium raymondi for fiber strength and fineness, and tomentosum is also for the same trait. Arboreum, herbaceum are the two cultivars of cultivated cotton. Tomato, in tomato Lycopersicum hirsutum for fruit color, carotene content. Lycopersicum pimpinellifolium for vitamin C, and Lycopersicum peruvianum is also for vitamin C. In pigeon pea, a Tylosia species, this is also considered as the wild ancestor of pigeon pea, which contributed for high protein content. In soybean, Glycine max is a cultivated species, and Glycine soja has high protein of about 45% protein, which reduced lipoxygenase activity. In oats, Avena sativa is a cultivated one, whereas the wild one is Avena sterilis, has high protein, about 27%. Coming to transgenic approach, a gene from any other organism, including species into which it is transferred, that is introduced into genome of an organism using a technique of genetic engineering in transgene. Transgenes provide powerful means of modification of quality traits. In order to use transgene efficiency and successfully, biosynthetic pathway, or at least key enzymes involved in biosynthesis leading to production of concerned trait should be known. Example, Lauricle in Brassica napus variety having high lauric acid, and Brassica napus variety producing hirudine, that is a antithrombin protein of high pharmaceutical value. Production of hirudine and similar other novel proteins or biochemicals in plants may be regarded as a novel quality traits which drastically changes the end use pattern of crop produce. The breeding approaches for quality traits. First one is evaluation of germplasm, mutagenesis, hybridization, interspecific hybridization, somaclonal variation, and genetic engineering. First one is screening the germplasm. This is important for such traits that are not obvious. Example, traits related to chemical composition and lysine germplasm lines in sorghum. Further breeding efforts will be required to combine the quality traits with good agronomic features or yield, since the germplasm line is expected to be inferior in yield and agronomic character. Mutagenesis, a desired quality trait may be present in spontaneous or induced mutant. Varieties released for quality through mutation in India, IIT 48, that is for thylline coccidio 3%, P721, that is Victus through DES and X-ray 30 kilorad direct selection in M2 generation, that is from T141. This has fine grain bitter test weight, better test weight composed compared with IR8, high lysine content and medium slender grain cooking quality. Jagannath was from USSR, mutant 428, that is Jolotisli for high cooking quality. Then in wheat, Sharbati Sonora, Pusa Larma, Stadler and Novosibirskari 67 from USSR for good baking quality. In barley, Hiplori, Notch 1, Notch 2, Riso 1508, Canor from EI. Then in Austria, Vienna, then Jetta Pellas. Pellas is pre-soaked seeds with X-ray treatment for stiff strain. Mary from Sweden for stiff straw. Christina from Sweden for good straw. This is selected from Domin into Mori. Bonneville 70 from USA for seed, that is gamma ray induced and for improved threshability. In Pigeon Pea, Vishakha 1 released from bark for fast neutrons, that was from T21 base material, 35% increased in seed yield. Co3 from Coimbatore, that is 0.6% EMS induced and the base material is Co1. Here bold seeded, high degree of shelling. TAT5 from Akola and bark, that is 1.5 kilorad fast neutron treatment to 1 T21 base material, and this has approximately 50% larger seed size. Then TAT5 that was a cross of Newton T22 into TT2 into TT8. TT2 was large seeded compact and TT8 is early, both induced by 2.5 kilorad fast neutrons to T21. This has medium large grains. In black gram, TAU1 for large seed size by gamma ray induction. In chickpea, NIFA 95 for high protein. In cowpea, CO5 by gamma rays nutritional value. Linola 989 in India for oil quality in linseed. In sunflower, Prevenets, that is chemically induced for oil content. In groundnut, Somnath, that's a mutant for seed size and oil content. Vikram BP1 and PP2 from bark by gamma rays induction for seed size. Sesamum, Sinai White 48 originated from Egypt, that was gamma ray irradiated for seed color. Suwon 155 by gamma rays for oil quality. Young beak from SA for high oil content and Ceodum for somewhat higher oleic acid. The third approach is hybridization. This is most widely used breeding approach to develop high yielding varieties with desirable quality traits. The breeding methods used to handle segregating generation derived from appropriate crosses depend mainly on the type of parents involved in the cross. If both the parents of cross are high yielding varieties having good agronomic features, pedigree method will be the most suitable. If one of the parents has inferior agronomic feature, backcross scheme will be the most appropriate. Only a limited number, that is 2 to 3 backcrosses may be made if the inferior parents has some desirable features as well. The segregating generations may be subjected to sib mating in place of selfing and selection in an effort to break undesirable linkages with the gene controlling quality traits. These considerations apply when quality trait is governed by oligogenes. Quality traits governed by polygenes may be improved by subjecting the segregating generations to a form of recurrent selection. Recurrent selection for 2 cycles increased seed protein content in rajma, that is French bean, from 21.9% to 24.6%. In soybean, there was an increase of 3.3% from 42.8 to 46.1% in seed protein content in response to 5 cycles of recurrent selection. This table represents a selected example of breeding for improving protein or lysine content. In French bean, by recurrent selection, 2.7% of increase in protein in 2 cycles. In soybean, protein content to 3.3% by 5 cycles of recurrent selection. In sorghum, by pedigree method, lysine content was improved. Barley, by backcross, lysine content. Sorghum, through mutagenesis, high lysine and the induced by diethyl sulfonate. Then in barley, high lysine by EMS and ethylene imine. Coming to interspecific hybridization, when individuals from two distinct species of the same genus were crossed. Wild relatives often contribute useful quality genes, example fruit color, fruit size, provitamin A and C content in tomato, etc. In many cases, the high quality lines derived from such crosses will usually serve as parents in hybridization program. It is unlikely that they will be used directly as varieties. The fifth one is somaclonal variation. The genetic variation present in tissue culture plant is somaclonal variation has been exploited for crop improvement, and several somaclonal variants are being used as commercial varieties. Example, scarlet variety of sweet potato has improved and more stable root color. Coming to breeding methods. Breeding methods used for improving the quality do not differ from breeding methods used for other characters. These are extensively used for improving quality traits like backcross, pedigree, single seed descent, recurrent selection, progeny selection, mutation breeding, somaclonal variation, etc. In common bean, that is Phaseolus vulgaris, pedigree, single seed descent and recurrent selection methods have been used for improving protein content. 2 cycles of recurrent selection increased seed protein from 21.9 to 24.6 in soybean, in sorghum also the same, which are discussed in the previous slides. In barley, backcross method was used to develop high lysine. High lysine line had small sized grain and low grain yield. In sunflower, seed oil content was increased from 30% to almost 50% by Russian breeders in 50 years using modified recurrent selection. In safflower, oil content was increased from 37 to 50% through reduction in hull content. In maize, seed oil content increased from 4.7 to 17% and protein from 10.9 to 23.5 after 70 cycles of recurrent selection in USA, but there was a dramatic reduction in grain yield. Mutation breeding has been used for development of high lysine lines in sorghum, barley and maize. Diethyl sulfonate, that is DES, has been used in sorghum, AMS and EI in barley for induction of high lysine content. Coming to the screening techniques for quality traits, breeding for enhanced nutritional quality involves lot of chemical analysis. Breeding material has to be screened for protein content, amino acid composition, seed oil content, fatty acid composition, vitamins and anti-nutritional factors. This requires close cooperation of biochemists. The selection of plants with better nutritional quality is done based on chemical analysis, which should be simple, cheap and rapid. Now, rapid chemical analysis methods are available for protein estimation, seed oil estimation, fatty acid and amino acid analysis. Coming to seed oil analysis. For oil analysis, it is done with the help of nuclear magnetic resonance, that is NMR, or nuclear infrared analysis, NIR. This is the non-destructive method of oil analysis. After oil analysis, the seeds can be used for sowing purpose. Several single plants in segregating population can be analyzed. This method is quite simple, highly accurate and very fast. By this method, 300 to 400 samples can be easily analyzed per day. For protein analysis, that is analyzed with the help of protein analyzer. The analysis is based on principle of infrared reflectance measurement. The seed is ground into flour which is used for analysis. This is very fast, uses 200 to 300 samples per day. Individual amino acid can be estimated by calorimetric method or microbiological method. Analysis of anti-nutritional factors is carried out by chemical method. The old methods of protein estimation like Jaldhal method, oil analysis with Soxhlet method were very much time consuming, though more accurate. Now fast methods have been developed. Coming to the concept of breeding for low toxic substances. In some grain legumes, oilseeds, vegetables, fruits and forage, toxic substances are found. These substances have adverse effect on human and animal health. Feeding of forage with a toxic substance will adversely affect the health of animals. Therefore, it is essential to develop varieties of forage and food crops with low level of toxic substances so that it should not have adverse effect when consumed by animals. Considerable progress has been made in the development of varieties with low toxin content in any crops. Breeding for reduced toxic substances require lot of chemical analysis. Hence, development is simple, cheap, rapid and reliable method of chemical analysis is essential. Here there is a list of anti-nutritional factors, type A and type B. In type A, protease inhibitors. Here these interfere with the human digestion process and nutrient utilization found in chickpea, pigeon pea, lima bean, kidney bean, peanut, cowpea, garden pea. Method to overcome by heat treatment, germination, fermentation. The second group is lectin or hemagglutinin. These agglutinate red blood cells and other type of cells, toxin to human when exceeds the limit. Sources are lentil, pea, soybean, kidney bean and peanut, overcome by traditional cooking and germination. Saponins. These give bitter taste, hemolyze red blood cells. The sources are alfalfa, soybean, French bean and Pisum sativum. Group B include phytic acid, gossypol and glucosinolates. The phytic acid leads to mineral insolubility by binding strongly with them, particularly with calcium, iron, magnesium, zinc and other trace elements. This is commonly found in soybean, faba bean, lentil, chickpea and phaseolus beans. Method to overcome are by germination, applying phytase enzyme. Next is gossypol. This binds with the iron and amino acids. The sources are cotton seed meal and the CaOH addition reduces the gossypol content. Glucosinolates cause legume goiter in children due to iodine deficiency. This is found in brassica, soybean and peanuts. Type C anti-nutritional factors are antivitamins. These decompose vitamins and many combine with them to form insoluble or unabsorbable complexes found in green beans, green peas, kidney bean, common bean, soya bean, pea and sprouts. Lethogens, and these antivitamins can be overcome by boiling or heating to a favorable period or frying. Lethargen. This is a neurotoxin or a paralysis of lower limb is a function of this, and it is found in Lathyrus species. The method to overcome is cooking in excess water around 90 degrees Celsius, draining off of the excess water, overnight cooking in cold water, steeping dehulled seeds in hot water, roasting seeds at 150 degrees Celsius for 20 minutes. Next is favic agents like favism, hemolytic anemia, and hemoglobinuria is caused by this and found in faba beans. Drying beans and cooking is a method to overcome this. Estrogenic factors that induce growth of feminine parts found in soybean, and this is overcome by germination. Toxic amino acids found in xanthol bean which lead to kidney malfunction. Anti-enzyme found in navy bean and its relative leads to amylose inhibition. Flatulence factors in soybean resist the digestion. Cyanogens in lima bean leads to human poisoning, mental confusion, muscle paralysis and respiratory diseases, and this can be overcome by cooking and discarding cooking and cooked water. Coming to the toxic substances found in different food and fodder crops. Kesari contains lethargen or neurotoxin bova, pigeon pea and cowpea has trypsin inhibitor, French bean hemagglutinin and trypsin amylose inhibitor, soya bean trypsin inhibitor and coitrogens, rapeseed and mustard erucic acid, eicosinic acid and glucosinolates, cotton gossypol, safflower polyphenolics, potato steroidal alkaloids, cucurbits cucurbitaceans, brinjal bitter principle, cassava CN glucosides, yam alkaloids, mango resins, field pea antivitamin E factor. In fodder crops, sorghum consists in the younger stage HCN content, and in the older stage it contains or seed form it has tannins and cyanogenic glucoside. Sudan grass has cyanogenic glucoside or HCN. White clover is also having the same. Alfalfa, saponin and plant estrogens. Sweet clover has coumarin and lespedez has tannins. Practical achievements in quality traits. Varieties with improved nutritional quality have been developed in several food crops in different countries. In common wheat, seed protein content, then in sunflower, seed oil content in USSR. In maize, seed oil content increased from 4.7 to 17% and protein content from 10.9 to 23.5%. In wheat, Atlas 66, important source of high protein. Many mutant forms or by different breeding techniques. There are several improvement in the quality traits, and in recent days there are many quality aspects which are improved in different crops, which are given in my other presentation or in other video. I have given the link in this description, you can watch it. Varieties with improved quality released in some crops in India, like in maize for high lysine content. Varieties are Proteina, Shakti, Ratan. Sugarcane, high sucrose, Co 671, Co 6806, Co 7314, Co 7704 and Co 62174. In barley for malting quality, Karan 15, Karan 92, Karan 280, Lathyrus for low neurotoxin, Osa 24. Soybean for high protein and high oil content, that is Lee, which has 43 to 45% protein and 23 to 25% oil. Rapeseed K88 with high oil content, that has 48.8% oil. Limitations of quality improvement. Most of the traits are governed by polygenes and polygens are controlled by the environmental factors or influenced by the environment, so their prediction and inheritance prediction is a difficult task. Difficult in estimate and evaluate, hence more financial requirement. Low heritability and affected by environment. Negative association of seed protein with grain yield in both cereals and pulses. In some cases, improvement in quality leads to reduction in grain size and yield. Example, in barley, selection for high lysine content causes reduction in grain size and grain yield. Sometimes quality character is found in wild relative or species that the crossability barriers will lead to the major problems. Analysis requires close cooperation of biochemists, which sometimes become limiting factor in the progress. Biotechnological tools have not been used widely in the crops. Thank you. I hope you liked this video and don't forget to subscribe my channel. Thanks once again.